Thanks. So good morning, everyone. Dan Ho in bioengineering here at Penn. So I've been asked to give a lecture, bootcamp lecture about fluids. As a matter of fact, I teach a course, undergraduate course for bioengineers about fluid mechanics or biofluid mechanics. So one semester course. So I can spend, the next, a couple of hours actually talking about, what a fluid is and the basic principles of fluid mechanics. But, to make this presentation discussion more interesting, so what I thought would like to do is to focus on one, specific kind of research topic, in fluids or fluid mechanics called microfluidics. Have you guys heard of microfluidics before? Yeah. So microfluidics, when people think about microfluidics, let me actually, So they often picture in their heads, those small-scale devices, right? So it's many people think that it's mainly about the fabricating or using those small-scale devices, but it's a whole lot more than that. So using those, small-scale devices, what happens essentially is that we gain the ability to control very small amounts of fluids in a very precise manner. So I'm showing here a couple of examples of microfabricated fluidic devices, or microfabricated devices designed to handle small amounts of fluids for a wide variety of applications, especially for biomedical applications. So this example shows these droplets, microscopic droplets, that can be formed in this, two-phase, microfluidic devices, and these droplets can be used as individual reaction chambers, for chemists, or we can actually culture cells in these, microscopic droplets for, PCR analysis and so on. This is another example developed by Steve Quake at Stanford, and consisting of multiple layers of elastomeric fluidic channels, and you can use actually pneumatic pressure to valve fluid flows, pump fluid flows for a wide variety of applications. And you might have heard about the company called Fluidigm. Actually, the Fluidigm company is actually based on this multi-layered salt lithography technology. And then, this is more of on the fundamental side, but we can actually flow multiple liquids right next to each other without any convective mixing. This is a very unique transport phenomenon that arises actually from the small scale of these devices. So, but anyway, just to kind of get the fundamental stuff out of the way, if I were to define what microfluidics is, actually, so this is George Whitesides. He's a chemist at Harvard, actually chemistry professor at Harvard, who basically started this whole field of microfluidics or biomicrofluidics. So according to him, he actually wrote a seminal review paper in Nature back in 2006. He defined microfluidics as the science and technology of systems that are designed to process or manipulate small amounts of fluids using channels, very small scale channels, basically. Okay, so this is a formal definition of microfluidics I would like to present to you today. Okay, then, so microfluidics, there's been tremendous progress in this technology, and as you can tell from this graph, there's been exponential increase in the number of papers, for example, patent applications focused on some type of microfluidic devices or technologies. So and most recently, let me actually just before I start my talk, I'd like to actually show you this slide about the current state of the art, of microfluidics, and so what's going on in this field right now. So Dave Beebe, shown here is Professor Dave Beebe. He's a biomedical engineering professor at the University of Wisconsin-Madison. He's one of the pioneers in our field. So he wrote a review article in Nature 4 years ago about the present and future of microfluidics. And in his paper, he actually had a section called the when microtest, stands for micro total analysis system. Okay, microtest technologies are the only solution to talk about really promising, unique opportunities for researchers in our field. So he actually laid out 3 specific areas of interest. The first one is developing, kind of microfluidics-based diagnostic tools for, resource-limited settings like, for global health applications. Another area is where, people use actually, microfluidics technology to develop, kind of clinical diagnostics, capable of handling, biofluids, for diagnostic applications. The third one is actually developing microfluidics-based models to mimic what happens in the human body. So I was fortunate enough to get deeply involved in pioneering this technology, the tissue on a chip, organ on a chip. You might have heard about these, terms. So what I'd like to do is, to make this kind of presentation more interesting, I'd like to actually talk a little bit about, what microfluidics, technology has to offer actually, for this particular purpose actually. So how can you use microfluidic devices designed to, handle very small amounts of fluids, to develop physiological, realistic models of human tissues and organs. So that's actually one specific focus, of my talk, today. Okay. Any questions so far? Okay. So this is a broad introduction, but. So let me actually start by telling you a little bit about the work that we do in my lab. Okay, so research in my lab basically revolves around developing these microfabricated devices that we use to grow human cells. Whenever possible, we try to use primary cells. You guys know the difference between primary cells and transformed cells? Yeah. So we actually use the cells taken directly from human subjects or patients, and then but we also use these devices to mimic the complexity of the human body, complex 3D structures, for example, dynamic environment like blood flow, tissue deformation, which is very relevant to what we're trying to do as a center here, at the CEMB. And so here, the idea is to use these microengineered, or the systems, as microengineered models of human organs or tissues, especially human organs. To be more exact, the functional units of human organs. Every organ can be reduced to a functional unit or units. Okay, so we try to mimic the structure, environment, and function of those functional units in our microengineered devices. So we call these systems human organs on chips, human. Some people use actually this term microphysiological systems. So what I'd like to do today is to give you a quick introduction to this technology and perhaps help you better understand what this technology allows us to do using a couple of examples of human organ on a chip devices we're developing in my lab. Okay, so these are specific areas of interest we're going to focus on. And again, many of these systems are designed for mechanobiology research actually. And whenever possible actually, we try to look at we have a lot of interest in modeling mechanically dynamic physiological systems like the lung, or the eye, or the placenta, where there's a lot of blood flow. So in those studies, we actually try to find out how those mechanical forces, physiological or pathological mechanical forces, affect the human tissues and organs. So that's we're going to see some of that, during my presentation. So let me start by talking a little bit about, microfluidic devices designed to mimic the human lungs. So you guys know how the human lung works, right? So the lung is a very complex organ, consisting of these functional units called the alveoli. So these are microscopic air sacs deep in the lung that expand and contract, as you can see in this animation, during respiration. And the most important function of the lung is gas exchange. And to make that happen, these air sacs are covered with capillary blood vessels. And then if you look at the tissue-tissue interface between these two compartments, you find a very thin layer of lung tissue, alveolar epithelial tissue on one side, capillary tissue on the other side, separated by a very, very thin interstitium. Okay, so the whole layer is less than 1 micron thick, which I find astounding if you think about it. Ultra thin air sacs expanding, contracting without rupturing all throughout life. I mean, that's really interesting. So when we actually started this project, we wanted to develop a lung on a chip, but essentially a microfluidic device that would allow us to mimic this essential structure, multi-layered tissue structure, and also, as an engineer, we found this very interesting, like dynamic mechanical microenvironment of the alveolar system in a tightly controlled environment. So that was our goal. So we actually developed this, system called a human breathing lung on a chip. You might have seen this photo somewhere. It became very popular. And then, this is actually a whole device. We use soft elastomer called PDMS, polydimethylsiloxane, It's a silicone rubber, basically, which is transparent, as you can tell from this photo. This material is also very flexible and gas permeable, which is great for cell culture. And if you look at the cross-section of the device, there are multiple chambers. So first of all, there are two chambers separated by a very thin porous membrane made of the same material. And so what we can do is we can culture lung cells and capillary cells on either side of the membrane to mimic the structure you saw. Another really interesting thing we can do is to apply cyclic vacuum suction to these hollow chambers. So what happens is actually in response to vacuum, these vertical walls, separating these two sets of chambers, these vertical walls are also very flexible. So when you apply vacuum, these vertical walls bow out, and that causes the deformation or stretching of the membrane as well as the cells and tissues, stuck to it. Okay? So by doing this, we can actually mimic kind of a physiological breathing motions in this microfluidic device. So just to give you a sense of scale, scale bar is 100 microns. These pentagons are pores on the membrane. Okay? So that are about 7 to 8 microns in diameter. So essentially what we do in this device is to culture these cells in a physiological, body-like environment. So, for example, we culture lung cells under air because that's the environment that these cells see in the body. And on the capillary side, we flow medium at high velocities to match the physiological levels of shear stress, hemodynamic shear stress, on this side. And so if you do that for like about a month or so, they actually differentiate. They change their morphology and function dramatically to become more like their in vivo counterparts. So then, as a result, at the end of the day, we can actually get this nice bilayer tissue that actually looks like the structural unit of the lung, alveolar capillary interface. But what's more interesting is that this guy also functions like the living human lung. So, let me actually give you an example. So think about lung infection as an example. So during infection, these bacterial cells, they're in the alveolar air sac, let's say that. And then what happens after that is these bacterial cells stimulate alveolar epithelial cells and other immune cells like alveolar macrophages to produce and secrete these pro-inflammatory cytokines and chemokines that are known to activate these capillary endothelial cells. Okay? Do you guys know how this process happens? So these cytokines, let me actually just jump to this, movie. So these, white cells actually, like neutrophils, circulating in the blood, patrolling the body. So they have this really interesting ability to sense those chemicals, and in response to that, what they do is they, start interacting with the activated endothelial tissue, and then eventually they squeeze themselves through the capillary tissue into the alveolar air sacs. And then once inside the air sacs, they start crawling. This is not how they crawl, but they start crawling directly towards the site of infection. So this process is driven by something called chemotaxis. There is a gradient of chemicals driving this motion. When they get there, they actually eat up these bacteria, phagocytose those bacterial cells to resolve the infection. Okay, so this is not tissue-specific, functional response. This is an organ-level response that integrates inputs from multiple tissue types, right? Very, very complex, response. The question here is, can this lung chip mimic this, right? So let's take a look. So this is the side view, the schematic diagram. This is the top-down view of the device. So these white dots are fluorescently tagged human neutrophils, white cells, flowing in the lower capillary channel. As you can see here, nothing interesting happens. They zip by, without doing anything interesting. But when we put bacterial cells on the lung surface to mimic infection, see what happens. These guys now stick, to this guy, because the endothelial cells are now activated. By activation, I mean they express these receptors, recognized by these white cells. What happens is these white cells bind to, those receptors, and that's how they start interacting with the endothelial tissue. How immediate is this response? So it happens within hours, actually. It's supposed to happen more rapidly than that, but yeah. So this movie shows the entire process. Neutrophil sticks, crawls around. At some point, it starts wiggling its way through the capillary cells and one of the pentagonal pores on the membrane to get. You can see this guy go out of focus because it's moving in the vertical direction from the bottom compartment to the upper chamber. So cells are everywhere, but they're not fluorescently labeled. Okay. And then once inside the air sacs, these white cells are shown in red here. They and these are bacterial cells. They chase the bacteria and phagocyte it. They eat up these bacterial cells to resolve the infection, just like what happens in the living human lung. Okay, so I guess the beauty of this is because of the optical clarity of the material that we use to fabricate those chips, you can see through multiple layers to visualize this very complex process in real time at high resolution. Which is very, yeah.
How did you culture the neutrophil?
So we didn't culture the neutrophil. We isolated them directly from human volunteers. In this case, it happened to me, and then so we labeled them right away. We of course fractionated them, labeled them fluorescently, and then put them into the device. Yeah. So neutrophils, especially those neutrophils, are very short-lived. So we have to use them like within 12 hours. So, which is one of the was one of the challenges. Okay, so I hope I'm doing okay in time. All right, so so that was actually a quick proof of concept, or not quick. Actually, it took me 3 years to publish this paper. So proof of concept study that we published in a major journal at the time, and I actually worked on this project as a postdoc, during my time at the Wyss Institute. So that was 8 years ago. So you might be wondering what's happening now, right, in the field. So one of the recent trends in the field has been to develop disease models, actually. So using these types of systems, we can start thinking about recapitulating very complex disease processes in the human body in these in vitro systems. So let me actually show you some examples of disease models we're developing in my lab here at Penn. Okay. So as you can tell from this slide, we're actually developing technology to study adverse health effects of cigarette smoking. So we're developing smoking lung on a chip, model to study how bad smoking is, or how okay, if it's okay to smoke. So in this case, our specific focus is on the distal, like the deep in the lung, the regions deep in the lung, because these are small-sized airways. Now there's very convincing evidence that these small-sized airways deep in the lung are the major sites of tissue injury and inflammation during smoking, but not much is known about what happens in this region, because it's hard to reach, actually. It's very difficult to get any samples from this deep region. It's very hard to image what happens, because we can't use bronchoscopy to actually access these distal region in the lung. So for those who are new to this, this is what the human airway tissue typically looks like. So there is actually the columnar epithelium. So it looks like a column. These are column of cells, and then this epithelial layers is supported by this connective tissue, stroma, containing stromal cells and vasculature, blood vessels. So to mimic this multi-layered architecture, this is a postdoc, now research associate in my lab, Mark Mandrinos, came up with this microfluidic device shown here. So it has 5 layers. First of all, to mimic the stromal tissue, what we do is we fill this middle chamber with ECM hydrogel, extracellular matrix hydrogel, along with the primary human lung fibroblasts. These are connective tissue cells found in the stroma of the lung. And then this layer is sandwiched between these 2 chambers, lined with airway epithelial cells and vascular cells. So using this device, we can culture them under air, for example, on the lung side, for 3 weeks or 4 weeks to fully differentiate them. So these are some specific differentiation markers. So these cells has become ciliated. They form these hair-like structures on the top surface. They also start beating. So the beta tubulin is a specific marker for those cilia. You can definitely see the cells express cilia, and they also form very tight barrier tissue. I'll just skip this. We can also form, it's not projecting very well, but we can embed these fibroblasts in the hydrogel, and these red cells are stromal connective cells, fibroblasts embedded in the gel. And so the starting material, the gel we use, is a type 1 collagen, but over time what happens is actually the cells start remodeling their surrounding matrices. They make and deposit their own ECM, such as fibronectin. So green actually shows a fibronectin deposition, by the cells. So this is how we culture the cells, but the more important question is how do we smoke them, right? That's a more important question. So this is how we do it. So we had an excellent undergrad in the lab. I think it's, let me mute it. So he and Mark actually came up with this setup. So it has multiple components. First of all, we use this cigarette smoking machine, computer controlled. We can actually generate, cigarette smoke in a very controllable fashion. We can mimic human smoking patterns. And then we humidified and diluted more than 96% to mimic very dilute concentrations of cigarette smoke deep in the lung. And then it gets delivered to the smoking lung on a chip in the downstream. So the important question is, what can we study, really? Yeah.
Cigarettes, did you go through any challenge of deciding what type of cigarette you was going to use, what brand, filtered, non-filtered,
Right.
menthol, blah, blah, blah?
Right, good question. So there is actually one particular type of cigarette that a lot of people use for research purposes. It's called the Kentucky cigarette. So in smoking research, actually, most people use those as cigarettes. So those are the ones we use in our experiments as well. But later I'm going to show a little bit. But we also tested electronic cigarettes. So there's a lot of interest in like the vapors. Yeah. So I'll briefly talk about that later. Okay, so these are some of the examples of kind of pathophysiological responses we can measure in this model. So have you guys heard of UPR? So it's well known that cigarette smoking disrupts proteostasis, homeostasis of protein. Like what that means is actually, if you smoke a lot, protein misfolding or unfolding happens more frequently. And when that happens, there's a cell organelle called an endoplasmic reticulum, which is responsible for fixing those and unfolded, misfolded protein molecules. When there are too many of these, dysfunctional protein molecules, ER gets stressed, and that leads to something called UPR, unfolded protein response. So this is a kind of a pathophysiological response, that people have suggested as a very important disease mechanism in a wide variety of diseases, including cancer, smoking-related, lung diseases, and so on. So this micrograph shows expression of these two UPR transcription factors by the epithelial cells before smoking. You hardly see anything, right? But if you smoke these cells, just one cigarette, see what happens. Within 16 hours, we see a dramatic upregulation of both of these markers very quickly. This is, again, as a result of smoking one cigarette. We saw the same thing in the expression of NRF2, which is involved in antioxidant response, the kind of stress response, before smoking and after smoking. Within 16 hours, we see increase. In this case, a translocation. These guys, when it's activated, it translocates into the cell nucleus. So that's what's happening here. So again, these are very, very early responses, but also they're reversible. So if we actually stop smoking these cells, they go back to normal, back to the baseline level. But in the longer term, what we found out is there are many interesting things happen, but one of the most interesting things that we found out is cigarette smoking, induces very interesting responses in this connective tissue, stromal tissue. So to remind you, this is what the tissue looks like on day 26 or something, of culture. So these cells are nicely embedded in the gel. They feel comfortable. They start remodeling their surrounding matrices. Green shows the cell deposited fibronectin. So when we smoke those cells on a regular basis, maybe one or two cigarettes, for 2 weeks, 3 weeks, 1 month, see what happens. So we saw a dramatic increase in both cell number, cell proliferation, as well as ECM deposition. So what do you think this is? Any guesses? Fibrosis, exactly, yeah. So this is a typical fibrotic response, what we see in a biological tissue. In many cases, we saw those very dense cellular nodules that almost resemble the fibrotic foci that clinicians see in fibrotic human lungs. So it was very, if you think about it, this is nothing new. Yes?
You also see muscle
Yeah.
also sort of seem to increase in individual cells. So have you looked at all of the cell contractility? Is it also increasing?
Yes, for sure, actually. So they actually become more contractile, and it actually often led to the failure of the gel because, they pull themselves and actually, and the gel actually pulls itself, and then it detached from the walls. And, yeah. Yeah, so we actually, didn't have a chance to measure the mechanical stiffness, but we're actually now trying to do that, but it's, from the immunostaining or histology, it seemed, substantially more, substantially stiffer. Yeah. You had a question? Yeah.
I'm not really that familiar with animal cell culture,
Yeah.
but been
Yeah.
monitoring and culturing them throughout this whole time? Because you said that at like 64 days things come back to normal, or like 48 days or something.
Yeah.
I might have misunderstood that one, but maybe to keep getting new cells, or is it the same cells?
Those are the same cells actually. So we kind of accumulate a bag of tricks in the lab to maintain those devices for long periods of time. Oftentimes the important requirements, for example, for submitting grants and stuff like that, we need to be able to maintain those devices for at least 2 or 3 weeks, and oftentimes 1 month. And so to meet those requirements, so we need to so we have to make them happy and viable before we do anything to the device. And so in this case, we culture those cells for 3 weeks, to fully differentiate them, and then we do smoking experiments, for varying amounts of time, but 1 day, 2 day, or sometimes 1 week, So you'd be surprised by the longevity of those models, and so. But always the question is the cells, So as long as you have the right cell type, the cells that are robust enough, you can actually maintain those devices for long periods of time. Yeah. Okay. Yes.
This might be a more general question. Is it possible to pinpoint what chemical in the cigarette is causing that? Nicotine, but then there's
Right.
thousands of other
Yeah.
chemicals. And then the second question is marijuana.
Yeah. Sure. Yeah. So to answer your first question, the short answer is no, we don't know yet. So as you pointed out, cigarette smoke is a very complex mixture of a lot of different things, and so right now we're just focusing on looking at the mainstream cigarette smoke, like like a diluted, but retaining all the original components. We do have plans to kind of look at the effects of individual components, but we haven't done that yet. Marijuana and other stuff, so we haven't tried, but we have tested electronic cigarettes, and we're actually seeing some interesting differences between regular tobacco cigarettes and electronic cigarettes. But yeah, there are a lot of interesting things we could do, including the ones you're suggesting. Yeah. Good? All right. So I was saying, this is nothing new if you think about it, because chronic smokers and COPD patients, commonly, often show, thickened airway walls and stiffened airway walls because of this. Okay, so it was interesting to see similar responses in our model. Okay, E6. So a lot of controversy, first of all. So we find it very interesting. So what we did was, we actually tested one of the electronic cigarette top brands, called Enjoy cigarettes. I'm not sure if you heard about it, but so long story short, let me actually quickly show you. So you already saw these. We saw some responses, but I'm not showing any quantification, but the response was reduced actually a little bit, okay, suggesting that maybe E6 are bad, still bad, but may not be as bad as regular tobacco cigarette or this particular brand. But again, we're actually trying to be very careful about drawing any conclusions from these experiments, because I know how critical or how sensitive these issues are. So one of the things to address those concerns is we can actually, get whole human lungs. We can do the same thing, in animals, but we can also get those human lungs from, we have a big, lung transplant center here at Penn. This is Ed Cantu is a thoracic surgeon working there. We are collaborating with. We can actually get those lungs, and then we can cannulate them to make those lungs breathe, and also we can perfuse the tissue to keep the cells alive for up to 24 hours. The only downside is actually those experiments are very expensive. It costs about $8,000 or $9,000 per experiment because of the expensive perfusate we have to use. But our plan is to use those ex vivo lungs to validate some of those in vitro findings. We're also conducting gene analysis actually to find out, because there are actually a battery of smoking genes that have been measured and detected in human lungs, and those data are publicly available. So we're actually conducting PCR analysis of those lung tissues that we smoke in the device for comparative studies, and so we're in the process of conducting those studies. Another example of the lung-related or lung disease models we're developing. So with the support from NIH, actually, we are developing a model to study asthma. So here, the idea is the same. We cultured 3 different types of primary human airway cells in this like realistic 3D kind of environment structure. So epithelial tissue, and we can form this gel. We can create the circular channel coated with endothelial cells to mimic vasculature. But what's really interesting, novel about this platform is we can apply compressed air into this hollow chamber to compress the entire 3D tissue in the lateral direction to mimic bronchoconstriction, during asthma attack. So long story short, we have a lot of data, but so what we're trying to do here is to use this platform to study how pathological mechanical forces, during asthma attack influences the progression and exacerbation of asthma. Because there's a very interesting clinical observation that suggests that bronchoconstriction alone, without any inflammation, okay? When people think of asthma, they think of it as an inflammatory disease, but regardless of inflammation, this bronchoconstriction alone can, exacerbate asthma. There's evidence for that, like clinical evidence. So but people don't know if that's really true or how that happens. We're actually trying to address those outstanding questions using this platform. So what we're finding out is actually, like when we culture normal cells and compress them, they don't really respond much, But if we culture, when we culture asthmatic cells and compress them, they become activated actually, and then they start producing more ECM. So there is evidence that asthmatic lungs are stiffer than normal lungs, and so we're actually finding a similar kind of response or phenotype in this model. Again, in the interest of time, let me just skip over this. Again, it's not projecting very well, but so last year we got this major award from Cancer Research Institute to develop cancer immunotherapy on a chip. Okay, so in general, recently my lab has developed this technique to microengineer those vessels. Those are actually, it looks much better on my computer, but so those red tubes are blood vessels we can microengineer in this device. And these vessels can be perfused with culture medium, cells, in this case, those 2-micron fluorescent microbeads. Okay, so what we do is we use this device. If you look at the bottom surface, there are these two structures that are sticking out, protrusions. And what we do is we can use this structure to trap liquid actually in the middle lane. So what happens is actually, if you inject liquid solution into the middle lane, what happens is that this solution nicely follows the middle lane without spilling over to the side lanes because it gets pinned actually, along these barriers due to surface tension. So it's called the capillary pinning effect. So we kind of leverage this principle to trap liquid solution. And this movie actually kind of demonstrates that concept. We're injecting black food coloring dye into the channel, and you don't see any spillage over to the side lanes, right? So what we do is we inject hydrogel solution actually containing these cells. We put endothelial cells and fibroblasts, and then once it's trapped, we put it in the oven to solidify the gel. And then after that, we take it out, hook it up to a pump, and then we flow medium. So what happens is actually all the stuff, good stuff, diffuses into the gel to keep the cells alive and happy for long periods of time. So over time, what happens is actually these endothelial cells, surprisingly enough, they self-assemble into these 3D vessels. Okay, so let me show you the movie. So this movie shows a self-assembly process over a period of 1 week. Again, I'm not sure if you can see them very well, but so we put fibroblast endothelial cells over time. These endothelial cells spread, proliferate, but they actually fuse and connect with one another to form this network of 3D vessels. The best part is they're also perfusable. See? So we're actually perfusing these vessels with fluorescent dye, 70 kilodalton fits a dextran. You definitely appreciate the three-dimensionality and also perfusability of the vessels. We can also perfuse them with larger objects like I think 10 micron beads in this case, or 5 micron beads. So now what we're trying to do is we can also make much thicker vascularized tissues. In this case, 2.5 millimeters from one end to the other, which is huge for a microengineered tissue. So now we're actually trying to incorporate 3D tissue structures like organoids, spheroids, and tissue explants derived from patients to develop more sophisticated vascularized human tissue models. So just to give you some examples, my lab is a part of HERN, Human Islet Research Network, sponsored by NIH. Along with my collaborators, we've developed this vascularized islet on a chip. So these are primary human islets. You can get those islets from human donors. You can put them in this device, and then vascularize them like this. Along with Patrick Sale and Wen Liang here, we are also developing human fat on a chip, vascularized fat on a chip. So back to the cancer immunotherapy project. So this is actually a tumor explant, lung cancer tumor explant that we got from a patient, and then we put them into our device to vascularize the tissue. So along with my collaborator Scott Worden at CHOP, we're actually developing a platform that we can use to, so the goal is to flow. Once we have this, tumor model, we flow leveraging the perfusibility of the blood vessels. We can flow blood-borne immune cells like neutrophils, macrophages, lymphocytes, and the goal is to study how they interact with the cancer cells in the tumor-specific microenvironment. And our ultimate goal is to use this platform to test drugs, especially immunotherapeutic drugs, designed for various types of cancer. So right now, the immunotherapy is really a hot research topic, as you might know. Traditionally, the strategy is to find the cancer cells and kill them directly, but our immune system has this amazing capacity to recognize them as threats and then kill them. But cancer cells are very smart. They have this special, interesting, the signaling pathways that allow them to hide themselves from the immune system. Okay, so our immune system can't recognize them right now. So the immunotherapeutic drugs are designed to get rid of, eliminate those, protective mechanisms so that immune system can recognize and directly kill the cancer cells. So this is actually really exciting and interesting, different paradigm, of cancer treatment. But anyway, so we're actually developing a technology, hoping that we might be able to contribute to, this collective effort, by providing a physiological and predictive drug testing platform. And in many of these studies, we're also developing ways to kind of scale up, the fabrication and operation, maintenance of those devices. Because organ chip devices are great because they can be made very complex. But the downside of that is it takes so much to make them, maintain them, do any experiments with them. So the low throughput has been a critical challenge, very major challenge in our field. So in this example, we're actually integrating a liquid handling robot to perform like cell handling or cell seeding or media replenishment steps in an automated or semi-automated manner. So this device only has 12 replicates, but we're also developing larger scale devices with more than 100 replicates on one system. So which I think is exciting. And most recently, my lab got selected to send those tissue chips up to space, actually, which is really exciting. So my grad student, Andre, took this fantastic photo using the space astronaut glove, that we got from NASA. So in this project with my co-PI, Scott Worden, we're actually trying to study immunosuppression in the microgravity environment. I'm not sure if you heard about this, but it's been known since the early days of space flight that the astronauts become more prone to infection or inflammation during or right after space flight. So our goal is to send array on a chip, bone marrow on a chip, devices up to the ISS, International Space Station, infect the area tissue. And what we're going to try to do is to mimic the whole process, entire process of immune cell mobilization from the bone marrow and recruitment to the infected tissue in the area on a chip device. That's because that's what happens when your tissue is infected. Bone marrow actually releases all these white cell neutrophils. They come to the infection site and they're the first responders. They try to resolve the infection. You saw the movie, so that's what happens. So we're going to try to actually model this process with the hope of finding out any interesting differences between unit gravity and microgravity. Okay, so that's our goal. We're scheduled to launch in February next year using the SpaceX 17 rocket, which is exciting. All right, so I hope I caught your attention. So I think this long-on-a-chip technology is really exciting, and I think it represents a major advance from traditional in vitro models. And so I'll skip over this, but actually MoMA, you can see those devices. So at MoMA long on a chip was formally acquired into their permanent collection. So for this, I got a lifetime membership from MoMA, which is really exciting. So I'm not sure if they still have this, but they have on the second floor they used to have this exhibit called "This is for Everyone," and at one of the corners of the room, they have 3 organ chip devices on display, and long on a chip is one of them, along with the liver and gut on a chip. So it was interesting that it was developed, for scientific purposes, but also designers found this model interesting. Yeah. So, but if you think about it, we can do this for any other organs, in the body, right? We can develop, skin, heart, kidney. So in our case, we had a lot of interest in the eye, actually, So more specifically, the front of the eye, called the ocular surface. So you can think of it as the window of the eye, responsible for protecting the entire ocular system from the external environment. So ocular surface has cornea at the center, surrounded by the conjunctiva. So the conjunctiva refers to this transparent tissue covering the white part of the eye. Conjunctiva is also found underneath your eyelids as well. And corneal tissue contains this stratified epithelium, contains multiple layers, supported by this collagen-rich stroma, connective tissue, containing these cells called the keratocytes. Conjunctiva epithelium contains these goblet cells that actually produce mucus, which is one of the key components of your tears. Okay. And from engineering standpoint, from mechanobiology standpoint, this is something we found very, very interesting. Blinking. Okay, we all blink. So blinking is critical for, as you can see in this animation, spreading the tear film, keeping the ocular surface hydrated. This is important because it lubricates the surface, and also it forms a very nice smooth surface for light refraction. So again, what we wanted to do is to develop a system to mimic those important, interesting features. So this is what we came up with, human blinking eye on a chip. So developed by, so this is actually CEMB student, Jung Yoon. She's actually participating in boot camp as well. She developed this really interesting system called human blinking eye on a chip. So this study is under review in Nature Medicine now. So this is what the entire device looks like. We use a 3D printing technology to make these dome-shaped cell culture scaffolds with the same curvature as a human cornea. And it's highly porous structure, as you can see here. So to mimic these tissue layers, we came up with ways to, I'm running out of time, but so, we came up with a way to put these, for example, keratocytes into the cavities of the scaffold along with collagen hydrogel. We also came up with the way to mimic this physiological, concentric tissue pattern, with corneal cells at the center surrounded by the conjunctival cells. So what we do is we form the tissue layer, we expose them to air again, to mimic that, kind of physiological environment. And as a result, cells differentiate. They change their function and morphology dramatically. So this is a scan electron micrograph of the scaffold before, culture. This is after 16 days of culture. You can definitely appreciate the fact that there's a tissue layer, and then the cavities are populated with the cells and hydrogel. So as a result of differentiation, the corneal cells that, they start off as a single layer, but they become stratified, forming 7 to 8 layers, as you can see in this section. They form tight junctions between them to form tight barrier tissue. They express their specific markers like cytokeratin 3 and 12. And this is, something we find, really interesting. So initially, their surfaces are flat, but as a result of differentiation, they form these beautiful microstructures like microplicae, microvilli, that are almost indistinguishable from those features, found on the surface of the human eye. Again, the conjunctival cells differentiate into those, goblet cells, mucus-producing cells, as you can see in the AB positive staining. So then what we do next is to combine this culture platform with this electromechanically actuated hydrogel. So we use a computer and to control this actuator. It actually slides back and forth, over the surface of scaffold. So this is a tear channel. We get the best artificial tear available out there. We inject it into the channel. We apply a little bit of pressure. It comes out. It gets picked up by the sliding eyelid and spread on the surface. So the reason why this is important is because this is the very mechanism, that allows for the formation of a thin film, tear film on the surface. So how do we know we are forming the film? So we actually used OCT, Optical Coherence Tomography. This is actually a clinically used instrument that clinicians, ophthalmologists use to measure tear fluid in human patients. So we measured the thickness. It was about 6 microns. The normal human physiological tear film thickness ranges from 3 all the way up to 7. So ours, falls within the physiological range. we did some modeling with Vivek Shenoy, who just gave a talk. And so with Vivek, we modeled the biomechanical microenvironment. So, for example, we can actually estimate a pressure exerted by the moving eyelid, blinking eyelid on the ocular surface. We can also estimate the deformation, slight deformation caused by the blinking eyelid. And surprisingly enough, people have measured those values, in the past, actually using very rudimentary, but very good, fairly good, apparatus. The measuring systems. And so if you compare those two, we saw like really striking similarities, kind of validating again the biomechanical microenvironment of our model. So then you might be wondering, are the cells okay? This could be very damaging to the cells. And so here we are showing the cells. Green cells are live cells. So cells remain adherent and viable even after repeated cycles of blinking. But it's more than that. So we're finding out that this is something I think it's directly relevant to our activities at the center. So we're finding out that the blinking helps the cells differentiate better. So if you compare those cells, cultured at the air-liquid interface without any blinking, the ones that have been blinked actually express much higher levels of their specific differentiation markers. So based on these data, we're speculating that the physiological mechanical forces generated by blinking might be contributing to the homeostasis of the tissue, maintenance, differentiation of these tissue, the cells. Okay. So now we're trying to turn this into a disease model, and we're focusing on one particular type of disease called the dry eye. It's not a fatal disease, but it's a very common the disease. And right now there are only 2 FDA-approved drugs out there for this disease. But it's a very complex disease, and there are many, many outstanding questions as to how this develops and how this progresses and so on. So we're hoping to use our platform to address some of those questions in the field. So the very first thing we had to do was to create a system that would allow us to culture cells at the relative humidity of room air without using the incubator, without killing the cells. So relative humidity of room air is about 40–50%. If you actually did this, just in ambient air environment, cells would dry up or die quickly. So we actually came up with a way to humidify, the environment, and then also, keep the environment more physiological. To mimic, dry eye, what we did was to reduce blinking frequency. This is like one of the most common causes of evaporative type of dry eye disease. So you might, have experienced this. And so if you look at computer monitor screen for too long, you realize that you don't get to blink as frequently. Okay, and if you do that for long periods of time, you end up developing evaporative, dry eye disease. So we are mimicking that. These are 2 core mechanisms of dry eye. So as you can imagine, water evaporates very quickly from the tear film, and that actually increases osmolarity of the tear film dramatically. It's called tear hyperosmolarity. And also evaporation makes the liquid layer unstable because it gets thinner and thinner, at some point it ruptures. It's called tear film instability. So first, we actually used a clinically used instrument called the tear lab to measure and compare osmolarity. We saw striking similarities between eye and the DED patients. This is a really cool emerging technology used in the clinic to check tear film stability called Kurata Graphi. And so the patient looks at the cone, and the ring patterns are projected directly on the surface of the ocular surface. And the built-in software can track these individual rings to detect any ripplings or rufflings, as an indicator of instability. And at the end of the day, you can measure something called tear film breakup time. Okay, so we use exactly the same instrument. Here we're showing that we can project the ring patterns on the surface, and we saw very significant differences between the normal device and DED device. I'll skip over this, but again, this is another great example kind of demonstrating the power of this technology. All right, just a quick mention. I only have a couple of minutes left, and so we are doing a lot of work in the area of reproductive biology and medicine. So this is actually an area, so there are various small. There aren't that many good tools in this field actually, and then doing any type of clinical studies on pregnant women, raises a lot of, almost impossible, raises a lot of ethical, logistical concerns, and so they desperately need better tools and better models, and so this is where our technology comes in. So with this vision in mind, we're actually focusing on developing reproductive organ models, especially female reproductive organs. We published multiple papers about the placenta on a chip, and we're also developing cervix on a chip. So in the interest of time, let me just quickly show you one summary slide here. So placenta on a chip device, actually placenta, there was a big New York Times article 2 years ago about how little we know about this amazing organ. So arguably, this is the least understood organ of the human body. So placenta actually, one of the most important functions of placenta is material exchange. So if you look at the placenta, so this is actually the whole placenta, and then there are these repeating structural units. And then if you look at this tissue there, then there is actually kind of similar to the lung, but on one side there is a tissue covered consisting of those trophoblast cells. Those are the cells facing the maternal blood. And then on the other side are the fetal capillaries. Okay, so and what happens is actually maternal blood flows into this space called the intervillous space. Actually, this is the maternal side. Maternal blood comes in, and then there's a pool of blood in this chamber. And then there are these like tree-like structures called the chorionic villi. Within this chorionic villi, you find these fetal capillaries. So there is an exchange of glucose, a wide variety of biomolecules, as well as the xenobiotics like drugs, between the maternal blood and fetal blood. And this is the barrier actually called the placental barrier that actually regulates, this material transfer process. So it's a very, very important unit. And then also, if you were to find out whether or not a certain drug, gets through, then you would have to form this tissue and use that tissue to test the material transfer. So we use this very simple device consisting of top and bottom chambers with the thin membrane in the middle. We got cells, those are two different cell types, culture them on either side of the membrane, very similar to the original long-on-chip system. And we use this platform to, first of all, to show that we can form this placental barrier-like structures. And then most recently, we also use this system to demonstrate physiological transfer of biomolecules like glucose and also drugs. We tested a drug called glyburide, which is commonly used for gestational diabetes in the clinic. So this drug is known to not to actually get across, because what happens is the maternal tissue takes up these drug molecules circulating in the blood, but they spit them back out actually, because they have efflux transporters, expressed on the surface. So we were able to simulate, that protective effect in our microfluidic device. But again, kind of relevant to our, center, we're finding out that fluid shear stress actually plays a very important role in regulating those processes. Okay, I'll skip over this. Sorry, it's too many. So just to sum up, this is what we do. I hope you enjoyed it. So we we're not married to microfluidics and microengineering, but again, kind of back to the main theme of my lecture, we make devices, that we can use to handle very small amounts of fluids, but it's a whole lot more than that, as you have seen. We can culture those cells, maintain those cells, and also form physiological tissues using the novel capabilities of microfluidic devices to handle very small amounts of fluids. And just to tell you a little bit about the impact, the original long-on-chip system completely changed the way government agencies thought about in vitro testing, and became one of the most important reasons why they started new funding programs. And Obama actually made an announcement highlighting those major plans 7 years ago. NIH actually started a new funding program called Tissue Chip Program to support new research activities in this area, which is exciting. We also started a company called Emulate, based up in Boston. We're also in the process of starting another company based on like a brand new technology we've developed here at Penn. So in partnership with pharmas and academic investigators, we're trying to turn this into a screening platform for biopharmaceutical applications. Many people think about drug discovery. They're very excited about the possibility of using our technology for drug discovery. So I gave a TEDx talk 2 or 3 years ago, I guess, to talk very specifically about what this technology has to offer to improve the current drug discovery pipeline. We wrote a similar review article for Nature Reviews in case you're interested. So with that, I'd like to thank my group, slightly outdated photo, my collaborators, and my funding sources. Thank you for your attention. Thanks.